Thank you very much. Thank you very much to all of our speakers. Some beautiful talks between some reviews of old data and some super exciting new directions in data. So we can open up the floor to the discussion right now. We'd like to do it in a pseudo uncontrolled way. So let's try and all of ourselves behave and let's jump in with questions. And if we see there's too many overlapping questions, maybe we'll use the hand raising button. But we can start just as an informal discussion.
I have a question.
Hi, Farouk, go ahead.
Was it fast enough?
Super fast.
So we talked about layers, and these layers were mostly based on the cell architecture, and we were trying to reason about the layer functional MRI. So my question is, at least from my limited knowledge of the literature, there's a missing gap with regards to the angio architecture of the layers. Like we know the cellular architecture of the layers, myelo architecture, and a few other architectures too, but not so much I see like angio architecture. And I would like to go back to the history that Kamil taught us about, which he said, watering a flower while generously sprinkling the surround. This is for the laminar activity, and why is it detectable versus watering the whole garden. So now my question is, if we assume that there are different types of angio architecture in different brain regions, would it be possible that both of these hypotheses are correct to a certain extent? And whether the relationship between the cellular architecture and the angio architecture is very tightly coupled.
Who are you addressing that?
Well, everyone. Maybe Sangji can like give some, if he knows something in the animal literature.
I think so. I can make a comment. I did a lot of optical imaging by myself, so I know what's the optical imaging about. And optical imaging tend to look at the surface cortex. So if you look at the surface cortex, then it's a large vessel is not well controlled. If we look at the layer study, and the deeper layer, obviously much better, smaller vessel, you're looking for small arterioles, capillary, they are much better controlled to the neural activity area rather than large vessel. So I think some more like a weighting of the which vessel are you looking for. So it's a fMRI, we have a layer, it's like a depth information. So we can have a much less weighting about the surface vessel. Optical imaging is a highly weighted surface vessel. So I think that's appear to be different there. I think that's what the major reason in optical imaging see the widespread response, and fMRI showed very well localized.
Mm-hmm. And now I would like to ask one step deeper into this comment. Imagine two areas, right? And imagine having one very dense angioarchitectonic layer in the middle versus another area, let's say frontal cortex of the animal, you have a dense angioarchitecture layer all throughout the gray matter.
Okay.
I think would you expect,
Yeah, I can make a comment, a couple of different things. Okay, they have a different area, will have a different angioarchitecture. All right, there are different capillary density, depending on area. Some layer have a more higher capillary density in other area. For example, I gave a talk about olfactory bulb. Olfactory bulb, there's upper cortical area called the glomerular area. There's a highest capillary density, and usually have a visual cortex area or somatosensory cortex area. Layer 4, there is input layer. There is a highest capillary density. Now, I think important issue is olfactory bulb. When we stimulate different layer, we do see a CBB response follow active layer, even though is a glomerular layer, the upper cortical layer, they have a highest blood volume, baseline blood volume, but activity is a follow where is a real neural activity there. So that's what I can say. Yes, there is a difference there, but it's a evoke response. So we are looking for evoke response. Is evoke response actually quite specific?
If I can make some self-advertisement. I have reviewed the literature on the vasculature. And even though layer 4 has in some areas higher density, the difference in baseline blood volume between the different layers is actually not as dramatic as one might think. So from the humans, what we know, V1 has, for example, layer 4 quite high density. It's 2.5%, but layer 3 and 5 have 2%, and that's already considered to be extreme, which means the layer, the vascular differences in baseline blood volume is actually not as high, except obviously layer 1 and layer 6 are special. However, when it comes to macrovasculature, which means like the draining vein, we have an increase of baseline blood volume in veins from layer 6 to layer 1 because more blood has to be drained out. So it accumulates. And the same for arteries, where you have arteries which are sort of ending in layer 2, arteries which are ending in layer 4. If you sum them up, you have higher baseline blood volume in the macrovasculature closer to the surface. And when
all
it
in
comes
V1,
to column,
you mean?
No,
is it for the whole brain or view or
we don't know about the whole brain because we don't have much data, but I would assume it's quite similar in everywhere where we have a microcircuit. When it comes to columns, there's no relationship of the sort of the areas which are perfused by big vessels and drained to the columnar organization. So, again, I reviewed the literature as far as we have data. There's not much data, but that would be the emerging picture when it comes to vasculature and lamina and columns.
Thank you.
Can I go one step deeper into it, or should we pass to someone else?
Let's see if someone else has something else to say, and then maybe we can resume with you, Farouk, later. Is there any other question? Because otherwise, I may have something, so I'm overriding you, Farouk. Yeah.
Go ahead, Prakash.
So first of all, I mean, there are differences within columns across species on the vasculature, but the hemodynamic signal ultimately comes from neural activity, whatever that means. So there are also differences in the spatial structure of neural maps across layers, across species, columns, non-columns, and that's going to obviously profoundly affect the kind of hemodynamic response you get. So it's useful to look and know about the angio architecture, but it's also important to know what the neural map structure is. So the other thing is, it was said that optical imaging is only superficial, and that is not really the case now. So that I just wanted to correct that. And the last thing from the first talk was the issue of the point spread function with different stimuli techniques. So in the olfactory bulb, the olfactory stimulus and then electrical stimulation in different areas. And then it was posed that it's a big unknown why it activates the input, why electrical stimulation activates the input layers. We do know the answer to that question, and there are papers to back it up, that when you do electrical stimulation, the synchronous nature of electrical stimulation engages local inhibitory neurons, and you shut off the rest of the cortex very easily. So you can only get the input activity, and then to say that a certain kind of hemodynamic signal is reflecting activity in a lamina, in a specific lamina, whether it's olfactory bulb or cortex, is quite misleading, because if you present a natural sensory stimulus, that would not necessarily be the case. Again, the very nature of electrical stimulation, almost anywhere in the brain, except maybe the spinal cord, engages feed-forward robust inhibition, and you shut down most of the circuit, other than the monosynaptic response.
So maybe I can make a comment about Prakash's comments there. Yes, I mean, olfactory bulb is a special model system. So when you do the electric stimulation, they have a synchronized activity in certain area. And the system is very specific there. I don't know that is involved is excitatory circuit in other area, and we do not know what exactly is other things going on. But what I, based on experiment, what I do in the mouse there, and what we are finding in the is olfactory bulb actually apply to the mouse too, because we do have like somatosensory stimulation. We do have a very well localized activity layer 2, 3 in especially like a primary motor cortical area. This is a relatively small area. So, I think there is obviously can be depending on, in real cortical area, they're a lot more complex than olfactory bulb. There is less control than olfactory bulb there, but I truly believe CBB is a very well localized to neural active site, whether this is 100 micron or 150 micron, whatever they are. But I believe it's 100 micron is my value what I'm looking for now.
Thank you.
I'd like to, Luca, can I ask a question?
Yeah, absolutely. I was just going to address it, yes.
Sanjay. So Sanjay, you showed with the CBV response that early time points you see CBV response in the in the outer layers, and then later on it actually shows up and dominates in layer 4. So, and then your image, the image you showed essentially was showing CBV response more or less in this middle layers of the cortex. So what has happened to the outer response? Is it has it disappeared, or is it just a thresholding effect that it doesn't show up in the image afterwards?
Okay, I think this is answer is reason is we do averaging of the all time. So initial first 10 seconds is a contribution relatively small compared to the remaining signal there. So I think it's an averaging effect. So if you do you only use the first 10 seconds, it's not going to be very good. But you use all the data set, then it's the initial first 10-second data does not contribute much. Just a sensitivity issue. Kamal, do you get it?
Yeah, I got it.
So what I can actually make a comment. This is actually a very important issue to the human fMRI community. I do believe. Reason is, when we, in the bold fMRI, we like to do very short stimulus, to detect capillary response. Now, when we use CBB, what should we do? Is a suggestion is, should we do very short stimulus, look at the initial signal, or you want to have a long stimulus, you want to see the later signal? That's what is a question you can ask, all right? So in the vessel, obviously, you cannot do a very fast experiment there, but based on what we do experiment, not just only to the layer, we also have a column data. We publish paper in the column data set. Initially, there is a penetrating artery will dilate first. Okay, so penetrating artery is dilated first. Obviously, small vessel may be dilated. I don't know, but then later on, then is a small vessel is a more likely, like maybe capillary, maybe it's a precapillary arterioles. They are dilating those one. So when we detect those signals give you layer specific signal or column specific signal. So we can do single like a condition map from the CBB, and we actually get that map is very good. But when we only use later time point, most optical imaging study, what the Greenbold actually is prosthetic, that paper say CBB is not a very good. Only good is a very early time point. Very early time point looks good, but later is not very good. Reason is, optical time scale is much shorter than is fMRI time scale. They only do a couple of seconds stimulus, then trying to look at the early time point. If you do the optical imaging, same thing. If you do 20 seconds, then later time point is much more localized than early time point. So this is more like a vascular regulation, I suppose.
So a clarification question on that. There's a change in the neural response to a long sustained visual stimulus, for example. So do you know whether the effects that you observe that are different over these time periods are due to changes in the neural response or due to changes in the vascular response?
I do not know exactly because we do not know exactly how the neural response changes because of the time there could be inhibitory neural active, then there is a more sharpening in the active site there. So there could be have a neural contribution, but there is definitely there is a large vessel versus small vessel contribution there too. So I could not eliminate the possibility about the neural contribution later on. They are much more sharpening. Tuned to those small area.
A different way of,
May I?
a different way of saying the same question is, we know that the neural activity adapts very quickly. So with these very prolonged stimuli, maybe 5 or 10 seconds is okay, but when you're going to 30, 40 seconds, you're studying hemodynamics that don't really map onto meaningful neural representations.
Well, I don't want to make a comment that on because I think I'm MR physicist. I'm looking at the more like because when you do that on true spiking wise, but you got a synaptic activity still coming in. You stimulate, you still see there is a adapt is very quickly true. There is still there remaining as a signal there. So I mean, I don't. It's yes true. If you do that, then you should do the very short stimulus.
And I think that, there is also the complication with the eye movement. So, I mean, I think that, even when we actually, think about like a 30-second stimulus, if you don't move your eyes, you don't see. What that means is that, we have the constant microsaccades coming in that, refresh. So there's all sorts of interesting physiological properties there, right? But I want to pick up on this because I am very excited and puzzled about the new results of Matthew Larkham, where they show these XOR properties in suborbital layers. So one even more complex, thing that they show is that if you present two stimuli that are actually full contrast, the neurons shut off, or there is no dendritic spiking, whereas they only respond when the stimuli are at threshold. So I think that, what probably one of the challenges that we have is that we need to also characterize what stimuli do to our responses and be much more careful as to what we are showing to the cortex, because it might not actually respond the same way and may not engage the neurons in the same way. And the responses might be not just because of adaptation, which is something that we know for long, but if these new responses are true, or if these new data are true, there is a lot more that is happening than we had, somehow taken care of.
So can I make one comment? Is so I think if we also do a lot of depth electrode, we record the electric activity across layer. So if we record NUA activity across layer, then we don't see much difference during the pulse stimulation. When you have a electric stimulation in the pulse, so essentially you're looking for some sensory cortical area. You put the 16-channel electrode. If you look at activity there, upper cortical area, granular cell layer, and the infragranular layer, there is not much difference. It's a mainly difference onset time. It's when you start the initial spike there, but it's just a number of spikes like that on, there is a very little difference. But when you look at the fMRI data set, we do see highest response middle cortex. There's a granular cell layer. So that means there is a mismatch between what your people talk about MUA, or like most of neuroscientists interested about the measurement there, then fMRI measurement, there are certain mismatch. So I think there is a more toward to us is a more like a synaptic activity. So where is a synaptic activity, you are not measuring it's not easy to measure those ones. So that's one is probably a major driving force for the fMRI signal. Somehow it's a hemodynamic response. That's what my guess. So
but again,
the one thing I would say again is with electrical stimulation, the nature of the stimulus quenches the cortical circuit in a way that you don't get with sensory stimuli, whatever the sensory stimulus is. And there are papers, including from my own lab, that show, for example, in mouse, that across cortical layers, if you use visual stimuli, there are differences in the magnitude of the response and the selectivity of the response across across layers.
Okay, I think it's, okay, I just want to go one step, and I'm not going to talk about much about those. We did a lot of visual stimulation studies. I don't think it's just whether electric stimulation pole is making so much matter. Yes, that is obviously not a natural stimulus, but we do the viscous study, we do see the same things. I think it's I think that's is more like a side issue, whether, you know, what fMRI measurement, what we measure is a little bit side issue. So it's I think the key point is CBB response appear to be specific to whatever synaptic input layer. That's what I found. And then somebody else showed something else. I'll take a look at those one.
Okay, so question for you, CBV and BOLD and CBF people. So I mean, BOLD only works because there's a mismatch between flow and metabolism. If there wasn't, you actually wouldn't get a BOLD change because the oxygenation and the flow would be well matched. So my question is, why does the CBF and CBV show the selectivity that BOLD doesn't? Is it because there's differential metabolism across the layers of the cortex, or is it only because of the draining vessel, the larger draining vessels anyways, that actually are obscuring the effect? Because, you know, in the absence of draining vessels, or if you go to very, very high resolution, I think this was Camille's point towards the end of his talk, if you could ignore those draining vessels, would you see BOLD just as specific as CBF or CBV?
For me, I will not do the BOLD for myself, actually. Yes, I think a major issue is BOLD is a mismatch between CBF and the CMRO2. So it's a middle cortical area. There is a high CBF change, high CBV change, also high CMRO2 change. So you are looking for mismatch. So you are going to be less conscious between middle cortical area versus upper cortical area, even if there is, or you only look at the capillary. All right, do you understand that? So I think it's just conscious law. And I did a lot of spin echo study by myself. I did a lot of fMRI study, spin echo, and I was not very convinced. You know, I can actually measure like an activity map, like what we see in CBF or CBB. So, you know, I'm not doing any human study. So human study, you don't have any option. So you have to stay on to spin echo or gradient echo and or vessel there. But in animal study, I prefer to go to the like a CBB study if I need to have a localization to the specific layer like the localization.
If I can add to that, I think that the answer to Ravi's question is we don't exactly know. And I think we don't exactly know the answer to that question because yes, we are showing some CBV responses or CBF responses that look more precise than BOLD responses. But we know that in the gradient echo BOLD responses, we have the big vessel confound, and we know that the spin echo responses tend to be more weighted towards the microvasculature. But the way we do those experiments, the way we acquire spin echo responses, they are always contaminated to a certain degree with gradient echo responses, and spin echo itself, has a little bit of maybe non-specific contribution as well. So it is in the BOLD we have this confound that comes from the vessel diameter distribution, and so the result I don't know of a single experiment where all of those effects have been addressed fully. And I think that some of the things that people are working towards will ultimately answer those questions. And I tend to believe that in biology, it's not just the brain. When you look at many organ systems, there's a really very, very tight coupling between all the parameters that we are discussing about CMRO2, vascular density, and blood flow. And the fact that we are looking at something different than, in BOLD than CBV or CBF, that is that also brings CMRO2 into the picture. It may be that we are still okay if we can actually suppress all the big vessel responses. But future, I think, will tell. There will be experiments, I'm sure, with very high resolution and with other techniques, etc., to answer that question. But we don't know the answer to your question now.
I agree with Camille. I mean, I think still we have to do more work to do.
So Sanjay, somehow I can feel the kind of like a filtering function using different part sequence of, CBV or different physiology parameters. Also, you showed a nice figure, even using CBV with the gradient echo and the spin echo sequence. Again, this depth, profile change again, if I remember correct. So when you use a gradient echo, you see a very beautiful peak at the deeper or like a layer 4. But on the other hand, if you use a spin echo, but also like a mine or like a contra aging, you start to see the tilt up for the, the super layer. So I think this is a more, address the Robbie question. And one is about the more oxygenation level. CBV, it looks about the volume change. So very like, even like a Prakash had a paper, they showed that for short stimulation, the venue actually no dilation, the significant dilation until very long stimulation. So then with CBV, maybe it's a good candidate, because look at the more arterial size, like a vasodilation. That's the perfusion regulation. So I think it really depends the sequence or what the physiology and even field strength. It's all kind of filtering function and the question which one give the best mapping with the specificity. Yeah.
Well, I think it's yes. I mean, all depending on what you're doing, and where is I think it's I agree. It's a lot of depending on what you're doing, but it's a fundamental physiology is there. So whether we can capture fundamental physiology properly, whether it's a technique is something is more sensitive to the large vessel or small vessel. It's whether oxygenation level or blood vessel dilation. So a lot of different physiology is happening. And there's a time-dependent manner, and we have a tool set. How you measure is those information we think is more close to the capillary area activity. So I think that's all. It's what we measure with what tool we have it. So if you, yeah. I think it's a way mentioned about it's a CBV study, spin echo versus gradient echo, their profile there. Their profile is actually gradient spin echo profile is really good. And those profile I show you is we actually calculate the relative blood volume change by correction of the bold component. So maybe is when we make a bold correction may not be accurate because it's a myon versus a bold. They are maybe not the same way we're seeing those. But anyhow, if we just look at the profile looks really beautiful. But it's a make a correction maybe more changes. I don't know. But I think as you said, there is all depending what the tool you're using.
Yes, Sanjay, maybe one kind of question relevant to the points. I think that we had a little bit conversation with Ben Tandy. He always asks, say the CBV weighted imaging versus the CBV relative change. Sometimes they see the two peaks, but after correction, maybe only see the one peak. So what's your opinion about that too? One is to correct the bold effect and another without the correction. But assume you use the large dose, like the contra aging, so kill the most bold signal. For that kind of condition, correction before or after, how we interpret the kind of results, look at it like a late dependence.
I think it's fMRI is an animal study. We inject the myon, it's quite large dose. I think mostly it's a CBV weighted images. It's a correction of the bold signal is relatively small. So you only change is somehow scale, but it's not change is mainly about the general shape. Okay. No, I think it's not going to change the general shape.
I think like it might be important to consider, if you consider the blood volume, the quantitative blood volume signal with respect to which baseline. If you consider it with respect to like the blood volume change in units of voxel volume, or blood volume change with respect to the baseline blood volume change. Like as Camille Uluda earlier already mentioned, the macrovascular response in the superficial layers is much bigger. So if you look at the relative blood volume change normalized to the baseline blood volume, the superficial peak has a larger denominator because you're normalizing with a larger baseline. Whereas, however, when you only look at the quantitative change of blood volume with respect to the voxel volume, like in units per milliliter, then you might end up with a slightly different shape, completely independent of the boat correction that Sonji talked about. Would you agree, Sonji?
Yeah, I think the unit is important. So I think one of the things we did is arterial blood volume measurement. This mainly is all using the ball technique with the different empty level. That's what I mentioned also. And in that measurement is we don't see much of a surface vessel. But I think all depending on what the unit, what you measure. So we have to be careful how you interpret data set.
Thank you.
If I may make a question. So regarding to the comment that Prakash did in the framework of electrical stimulation of neural tissue. So when we stimulate like electrocortical stimulation, we see like these visual lesions. So there are areas of the cortex that affect a specific cognitive function, for example, speech arrest and so on. So how will you reconciliate, for example, the phenomenon of getting a specific a perceptual phenomena, like for example, instead of losing the ability of gesticulation, you get, for example, reactivation of a voice or a song or something like that. So how will you reconcile these two with the shutdown of the volume that is being stimulated during?
I do not know exactly what is or the question about electric stimulation. Now, so okay, I can just tell you our electric stimulation. What we did not stimulate the cortical area. We stimulate the fibers, a fiber bundle coming into the cortex. As coming into the it's a olfactory bulb to cortical area. That's a lateral olfactory tract. That's actually white matter bundle there. And the other one is we also is anterior commissioner. This is also white matter bundle. So we actually stimulate white matter bundle in the sense. So it's not likely same as you do the electric stimulation is the white matter brain area. You stimulate the inhibitory neural activity and excited neural activity. You activate everything there. So you mess up the whole circuit there. And this is quite specific. We know is based on literature. Also we measure. This is activate certain specific circuit level. Obviously, there is a problem activate also brain too, but we don't measure brain, so we don't really care about the brain part there. We mainly care about the olfactory bulb there. Now, the other one is we keep confusing about the pulse stimulation. Pulse stimulation, we actually do the electric stimulation in the pulse. So it's more like a sensory stimulation. So it's not likely same as you put in the electrode in the brain to the microstimulation in the brain to you activate, you make a seizure, etc. Those. So I think it's all depending on how you do it and something you could control it there. So, and I think what our observation consists of whatever we measure is everything. We do have, it's a visual stimulation. We have olfactory bulb, we have study. So we have a lot of different studies internally consistent. So I don't feel like this is actually cause a problem for us how you interpret data set. So that's all I can say. And I don't know other people's work very well, and I don't want to make a comment what the general electric stimulation problem in the cortical area.
So I can say something about Mario's question. I don't know the human stimulation literature, but I do know that the functional domains, assuming columnar architecture in the human as it is in most rodents, but in humans, functional domains are infinitely larger than they might be in a mouse or a cat. And in many mouse cortical areas, there's no functional architecture. So having very large chunks of tissue that do the same thing makes it very easy to get a very crystallized, functional correlate from stimulating and getting a very clear effect. So that is one obvious answer, but I'd need to look at the paper to see exactly how it was stimulated. And I don't know much about the olfactory bulb, and I wasn't trying to bash electrical stimulation, but this is the reason why optogenetic stimulation is so powerful. And certainly in the cortex, it doesn't really matter whether you stimulate the white matter or intracortically. You get synchronous activation and you shut down the cortex other than the times that you're giving the electrical stimuli. So, and then with the human studies, it will depend on how you stimulate. Is it high frequency, low frequency, and so on. So there are a couple of reasons, and I don't know what study you're referring to and how it was done, but there can be some logical explanations for those results that you described.
So just to clarify,
I think we do a lot of go ahead.
Just to clarify,
Can I respond?
Yes, please.
Go ahead. So I think it's what we just have to do more study, and we do a lot of optogenetic study now. We look at the layer study, and I think it's it's to me like it's time will tell. So we have other people study, and as long as we have a cumulative idea, then we will have a better answer at this point. And I don't want to say this is the only way to do, and there is not. At that time we need to localize certain layer, and that's the way to do. So, but I think it's a time will tell you. All right? We do a lot of study now. We are trying to control different layers, doing the optogenetic stimulation, and we'll find out sooner or later.
Can I have another chance to ask a little thing? Or
I think Luca was muted.
Yeah, I was actually talking, but I was muted. Sorry, Farouk.
Sorry.
Okay, if it's very quick, just because it's getting very late, especially in South Korea. So if you have a very quick question.
Yeah, very quick. So I think I asked this question a couple of times, and Kamil Uludağ always gives me the same answer, but I think there's a misunderstanding between us in terms of the terms that we are using. I'm asking about angio architecture, and this is what I mean. So this is the earliest work that I could find about the holistic angio architecture mapping of an animal, a macaque monkey, from 1940. And you can see that there is a dense mid-layer going on here, let's say, and then it's getting different. And there are also other types of laminar angio architectures, like uniform. So my question was, do we need an angio architecture map to test whether this watering the garden this way or that way hypothesis might still be both true in different laminar angio architectures across different areas?
So in columns, the Pablo Blinda has shown that the single penetrating artery supplies more than a single barrel. So in that sense, if we have a vasodilation in a penetrating artery, it's a little bit unspecific to the column itself. But the microvasculature obviously is then much more selective. And if you have a weighted average, then you have still selectivity of the blood volume. And the CBF would be more selective because you get most of the signal from microvasculature. But if it's a macro and micro weighting, then it's still, I think, lamina and columnar weighted, but it's not pure lamina and columns. Is this answering your question?
I need to think about it. I think I didn't fully understand, but we'll think. Thank you. Thank you.
Thanks, Camille. Thanks, Farouk. And I just wanted to conclude quickly, maybe with a comment stroke question for all our speakers to kind of like bring the conclusion back together. And then we can let Professor King go back to sleep. Yeah, it's past 12:30.
That's okay. I can answer those. You know, it's exciting those.
Thank you. But I guess the question is like, so we've seen across the three talks, there's some very exciting landscapes for laminar fMRI, and some we've seen a good degree, well, a degree of correspondence between electrophysiology in animals and humans and laminar fMRI. And when we have, but obviously, you know, we know there's some limitations across sequences, across approaches, across analytical strategies, mainly focusing around the sensitivity and specificity, and in both in times and in the temporal domain and spatial domain. And obviously, once we have like specific animal models against which to compare our laminar results in humans using fMRI, it's all easy. You know, we know what's real, what's not real. But as like one of the advantages of doing obviously laminar fMRI is asking questions that we cannot ask in animals. We investigate, for example, higher level cognition stuff, and obviously we don't have a ground truth and we don't know what to expect. And likely, depending on the approach that we're going to use, we're going to get different results. So the question is, how do we know what's real and what's not? And is there like some suggestions to really evaluate the reliability of our results with fMRI when we don't have a ground truth? I know there's no answer to that.
Well, Luca, but if I understood you correctly, but you did say that we can have ground truth in in animal models, and in the animal models, and you can get to very complex animal models, namely in the non-human primates, you can approach in many areas, not every area in the brain, to a ground truth, if you like, by doing the kind of studies that Lucia mentioned, or optical imaging studies and electrophysiology studies. So I think there is, and in addition, of course, in the human, very rare situation, but you do have surgical cases where you can also do the kind of experiments that that was presented. So in principle, you know, there are ways that a lot of people are pursuing to get to the ground truth. And ultimately, I think we can answer quite a lot of the uncertainties about what we are measuring with, for example, in the human data using BOLD or VASO, and you know, get answers that are you know, conducive to human neuroscience at the level of layers and columns. Another comment I wanted to make, I mean, we are always, you know, I think especially Farouk has been asking a lot of questions about, oh, what about the vascular density and, you know, vascular architecture, etc. But let us also remember that we are always looking at differences with respect to states, and we are looking at some sort of a response. So the baseline images that you get will actually reflect the vascular density to a large extent, but it is still possible, even when the vascular density is non-uniform, to be able to get responses that actually tell you something about at different parts of the brain what is going on. So we have to keep that in mind as well.
May I add to that? So I you know, that's why I try to put a lot of emphasis on why it's important to think about evolution, and, you know, like the different species that, you know, there are around, and which animal models might or might not necessarily be interesting for the question at hand. And I think that, you know, this is just not only homework that, you know, we need to do in the human brain, but, you know, maybe actually looking and exploiting non-primates will really be an important validation step. Now, are we going to address all of the questions? You know, when you ask like about ground truth, I think that it's probably going to get as ground truth as it can be. You know, so I probably over the years I've become a little bit more you know, more realistic in the sense of, you know, like whenever I look at because I'm interested in non-human cognition, then the alternative that you have is like whatever, like EEG, I mean, it's not that, like you are going to get very satisfactory answers either, right? And then, you get, like something like ECOG, which I love, and, like I do all the time, but then you always get this question like, well, but these are epileptic patients. How much does this reflect, human normal neurotypical cognition, right? So I think that eventually, we just try to look at whatever the brain does through the lens of, we know very little, and we're just trying to find out. I think that's the way in which I, like very pragmatically think about it. But I encourage us to think, because I know that there is many challenges on the laminar, and there is, and I myself, I'm more on the, like I don't really know what to make of these, results. But at the same time, I do see the importance, and I think that as we, like get data from the Allen Institute, we get more human tissue and we compare it, we do transcriptomics, we're going to find out that the human brain in many ways is actually an interesting beast, and so probably we're going to have to resort only to, like layer-fMRI, and it's not going to be, it's not that we have many more alternatives, so I think that it's also good to have that reality check, so just from the practical point of view.
I would like to make a short comment on stimulation, because human literature usually people use longer stimulation and animal usually shorter. And I think when we look at naturalistic stimulus, we have many different time scales. If I, for example, walk and focus somewhere in the far, then my visual field does not change much for a long time. So that would be like a continuous stimulation. If I have saccades, if I move my head, if a car is driving, we have something fast changing, but also not on the milliscale time scale. So I think naturalistic stimuli has the entire range of stimulations, and to play sort of the short stimulation versus long stimulation and declare one as a gold standard, I think does not correspond to the ecological validity.
I will even add, like to that, we did beautiful studies, where you look at blinks, because when you think that you actually have a 12-second stimulus, guess what? Actually, you don't, because you blink. So you lose the image, it is just much more complicated, like let's face it.
So you don't agree that there we have different time scales?
Well, no, what I think is, it becomes also interesting because the brain has figured out these ways, which is kind of like, creates either priors that, make us not to perceive that, we lost the sensory stimulation, and we nonetheless think that there is, sensory stability, right? But also ways by which probably it creates boosts of excitation, like through microsaccades, for instance, right? So I think that it's just, or put it differently, what I would contest from you, Camille, is that I think that the what we call naturalistic is probably a misnomer, because even if you just look at an image for 20 seconds, there is natural behavior that occurs. Now, is it nicer to look at, like a more naturalistic images? Probably yes, because the spatial frequencies and all the things that are in those are interesting. So we gain from that. But I would just try to say that even if we just look for 12 seconds, it's not that simple. That's the,
I didn't say it's simple.
I think your point about naturalistic stimuli is fine, and it's not about, when we talk about the duration of the stimulus, it was brought up because of the type of stimuli that was being used. When you use naturalistic stimuli, I meant like gratings, which was typical of what has been used in animal studies on neurovascular coupling. So with natural stimuli, the dynamics of firing rate is completely different. You get bouts of high firing and then long bouts of no firing in a given neuron or in a given area. But with a grading stimulus in a column, you're getting persistent activity. So it's not so straightforward as just the stimulus, but I was referring to when you have a given type of stimulus that has traditionally been used in animal studies, gratings or patches or whatever, then you can shut down the circuit very easily. It's beyond adaptation, microsaccades, all of these things interplay, but the dynamics of firing is very different in free viewing, driving the car, looking at the jungle versus looking at vertical lines and horizontal lines drifting across the screen.
I think we have to distinguish the sort of the methods level and the neuroscience level. On the methods level, you assume you can have any neural activity as long as the neurovascular coupling is sort of intact. And then you ask the question, what's the relationship between blood flow, blood volume, and Bolt signal? And that's what Sonji is doing. The other question is whether then the neuroscience activation pattern is generalizable to other stimuli. And then the answer for me is it depends. It depends on your question.
Well,
you can also have sort of a strong feed-forward stimulus in naturalistic stimuli and shutting down processing. That's sort of what happens if you have, say, conflicting information coming to one brain area and then it's not processed any further. So what I'm only trying to argue when people argue we should not do too simplistic, that now people sort of try to define their own territory as the only right territory and everything else is sort of delegitimized. And I think that's, we should not do that. We should sort of understand that there are many different questions to be asked and there might be the optimal method for the question to be asked, but not that the other questions are not legitimate to ask.
Yeah, thanks, Camille. And that's exactly the one of our main overarching goal of this meeting, trying to bring the flow together to come up with like the right way to do things together as opposed to my way versus your way. So it was a perfect way of ending the meeting. And I want to thank everyone, all the speakers and the audience, the floor audience, for being super well behaved and minimized mine and Renzo's work as moderator. In fact, it made it null completely. And yeah, thanks again to the beautiful, maybe Lucia wants to add something quickly.
Yes, I want to thank you and Renzo, because you are the guys that brought us all together. So, thank you for being such a good moderators.
It was easy when the speakers are so good. The attendance is just skyrocketing while you guys were speaking. So thank you so much. Thank you to all the speakers and all the floor. And yeah, hopefully we'll get to do it again very soon. That's our aim. So thank you to everyone, and we can finally let Professor King go back to sleep. Thank you so much.
Thanks, everyone.
Thank you.
Bye-bye.
Bye-bye.